Let's start looking at our genetic material. We first learned about DNA structure through three main experiments. Avery, MacLeod, and Marty in 1944 isolated DNA from Griffith's transformation experiment. Hershey and Chase in 1952 did an experiment with virus and bacteria showing DNA, not proteins, is genetic material. Watson and Crick published work showing structure of DNA. The structure of a deoxyribonucleic acid molecule is based on the pairing of nucleotides along the lengths of two complementary DNA strands, each of which has a sugar phosphate backbone and twists to form a double helix. Sugar phosphate backbone of each DNA strand is a repeating chain of the 5-carbon sugar, deoxyribose, and a phosphate group composed of a phosphorus atom surrounded by 4 oxygen atoms. The sugar phosphate chains are held together by covalent bonds that are generally only broken or formed by enzymes. Nitrogenous bases are attached to each sugar and is one of the 4 nitrogenous bases composed of carbon and nitrogen rings. Purines consist of 2 rings and are adenine and guanine. Pyrimidines consists of single rings and are thymine and cytosine. During replication, each strand of a DNA molecule acts as a template for the synthesis of the other strand, and when errors do occur, proofreading and repair mechanisms keep mutation rates low. Base pairing involves the formation of hydrogen bonds between the bases that are located toward the center of each DNA molecule, and they serve to hold the 2 strands together. Each strand acts as a template for the synthesis of its complementary strand through the addition of nucleotides to the growing end of the complement. DNA replication begins at origins of replication distributed at many sites along the length of each eukaryotic chromosome and usually at a single site on the chromosome of prokaryotes. DNA polymerase is the enzyme that catalyzes the addition of nucleotides to the ends of a growing strand of DNA through a process called elongation. The process of DNA replication occurs as follows. Enzymes, called helicases, unwind the helix at the origins of replication and help break the hydrogen bonds holding the strands together, creating a replication fork. Leading strand is the daughter strand that is synthesized into the replication fork. This strand is synthesized in a continuous fashion. Lagging strand is the daughter strand that is synthesized away from the replication fork. This strand is synthesized in a discontinuous fashion or in fragments called Okazaki fragments. An enzyme called primase then synthesizes a short segment of RNA called the primer that is complementary to nucleotides on the DNA strand. DNA polymerases bind to each separated strand and begin adding the proper complementary nucleotides to the primer to produce a new copy of each strand. Another enzyme, called DNA ligase, helps join the Okazaki fragments into a single strand. DNA polymerases keep moving along the strands until synthesis of both strands is completed. Each new molecule of DNA consists of one of the original DNA strands hydrogen bonded to its newly synthesized complement. Proofreading during DNA replication and repair of damaged DNA results in low mutation rates at the nucleotide level. Ribonucleic acid structure is similar to DNA in some ways, but has very important differences. RNA and DNA are both composed of nucleotides and both have strands consisting of a sugar phosphate backbone with nitrogenous bases attached to their sugars. RNA and DNA differ in the following significant ways. The 5-carbon sugar in RNA's backbone is ribose instead of deoxyribose. Instead of thymine, RNA uses the pyrimidine base, uracil. RNA is single-stranded as opposed to double. Adenine pairs with uracil and guanine pairs with cytosine. Transcription is when RNA is made from DNA. Transcription has 3 basic steps and occurs in the nucleus of eukaryotes and the cytoplasm of prokaryotes. During initiation, the enzyme RNA polymerase binds to the promoter and opens a portion of the gene to create a transcription bubble. RNA polymerase then begins to synthesize an RNA transcript complementary to only one strand of DNA called the template. During elongation, RNA polymerase moves along the template DNA, adding nucleotides to the elongating strand of RNA. Each incoming nucleotide base pairs with its complementary pyrimidine or purine in the DNA template sequence on the gene. During termination, a stop codon causes RNA polymerase to leave promoter and transcription stops. Translation is when mRNA is used to create proteins and it occurs on a ribosome. Synthesis of protein from mRNA occurs in the 5' to 3' direction. The process of translation has 3 major steps. During initiation, the start codon on the mRNA transcript is recognized by the ribosome. AtRNA attaches to the ribosome at one of the sites and its anticodon hydrogen bonds to the start codon. During elongation, the entire ribosome translocates down another 3 bases and reads another codon sequence, where another tRNA brings in the appropriate amino acid. A peptide bond between the amino acids is formed via an enzymatic reaction promoted by the rRNA portion of the ribosome. During termination, one of the stop codons is reached and the protein is released from the ribosome. Let's discuss the cell cycle. The 2 main phases of the cell cycle are interphase and cell division. Interphase is where the cells spend most of their time. G1 is the first phase of interphase during which the new cell grows to mature size and may begin to carry out its specific function. S is the second phase of interphase during which it duplicates its chromosomes by the process of DNA replication. G2 is the second phase in which it prepares for cell division. Lastly, G0 is when cells do not divide. G0 may sometimes happen right after G11. The second main phase of the cell cycle is cell division, which includes mitosis and cytokinesis. Mitosis is the process of cell division. It has 4 main stages. Prophase. During this time the chromatids condense. The nuclear membrane breaks down and disappears. A cytoskeletal structure called the mitotic spindle forms, and is used to pull the chromatids apart to either pole of the cell. 2 centrosomes are synthesized. In animal cells, the centrosomes contain small cylindrical bodies called centrioles. The centrosomes move to opposite sides of the cell. Spindle fibers, made of microtubules, radiate outward from the centrosomes and some, called kinetochore fibers, attach to each chromosome at its centromere. Metaphase. During metaphase, the chromatids line up across the center of the cell called the metaphase plate. Anaphase. Chromatids separate from one another during anaphase, at which point each chromatid is now considered to be an individual chromosome. The centromeres of the chromatids split. Telophase, the last stage of mitosis during which the mitotic spindle disassembles and a new nuclear membrane forms and surrounds each new complete set of chromosomes. Cytokinesis is the division of the cytoplasm following mitosis. In animal cells, a special collection of microfilaments of the cytoskeleton form a cleavage furrow in the center of the cell, which causes the cell to be pinched into 2 cells. In plant cells, vesicles from the Golgi apparatus form a cell plate in the center of the cell along which new cell wall material is deposited between the 2 newly forming plasma membranes. The cell cycle is regulated in order to help prevent the production of abnormal cells that could eventually become cancerous. Checkpoints are essential points during the cell cycle that regulate the process of passing from one stage to the next. Growth factor is a protein or hormone that promotes the division of cells. Density-dependent inhibition is the process in which cells stop dividing when they are in contact with each other. When regulation of the cell cycle does not work, cells become cancerous and keep reproducing without stopping. Asexual reproduction is a form of reproduction not requiring meiosis or fertilization. It only passes a copy of genes to its progeny. It's a type of reproduction in which there is no variation in genetic makeup. An example of this would be bacteria who reproduce via asexual reproduction. Sexual reproduction is a type of reproduction that involves variation because 2 parents give rise to their progeny. This introduces evolutionary advantage because of genetic variation. Meiosis, like mitosis, is preceded by replication of chromosomes. Meiosis 1 is the division where homologous pairs of chromosomes are separated from one another into 2 cells that are haploid, and it can be divided into 4 stages. Prophase 1, tetrads are formed or the pairing of homologous chromosomes via synapsis, chiasmata or the site of crossing over or exchange of genetic material is formed during this phase. Metaphase 1 homologous chromosomes pair with each other at the metaphase plate. Anaphase 1 homologous chromosomes separate and sister chromatids stay together. Telophase 1 the movement of chromosomes to the poles is completed. Meiosis 2 occurs in each of the 2 new cells after meiosis I is completed. No DNA replication occurs between meiosis I and meiosis 2. Meiosis 2 has 4 stages. Prophase 2, the chromosomes are already condensed and new spindle fibers form. Metaphase 2, each pair of chromatids lines up in the middle of the cell, and kinetochore fibers attach to the centromeres of each pair. Anaphase 2, the centromeres holding the chromatids together split, and one chromatid moves to each side of the cell. Telophase 2 nuclear envelopes reform around the chromosomes. Cytokinesis follows telophase 2, resulting in gametes. If sperm are produced, meiosis usually produces 4 sperm cells. If ova are produced, meiosis often produces a single egg cell, while the other 3 cells die or have other functions in reproduction. Mitosis produces diploid identical cells that have no genetic variation. Meiosis produces gametes, haploid, that are genetically different because of crossing over in prophase I of meiosis. Thanks for watching. Please don't forget to like, comment, and subscribe.